In today's video, we're going to be following along this wild-type developmental network. Reading from left to right, we have the protein S transcribing P, which inhibits I, which inhibits K, which transcribes E, which then transcribes the developmental gene of spikes on a dragon. You can think of the inhibition signal as a stop sign, meaning it will not transcribe the protein to the right of it, and the arrow or transcription signal as a green light in the traffic light, but we'll get more into detail later. To understand how each protein in this pathway promotes or inhibits the transcription of the spike gene, we're going to look at different scenarios. In the first case, the P has been removed from the network, and we are going to determine if spikes will or will not be transcribed. Going from left to right, S would normally transcribe the P protein, but since it is missing, will not do so. With the P gone, it cannot inhibit I, thus I is transcribed. I will then inhibit K. Think of a stop sign preventing the K from transcribing, and since K is not transcribed, E is not either, and thus we end up with a dragon with no spikes like Toothless. In the second case, I has been removed from the network pathway. S will transcribe the P protein, which will inhibit I, but it is missing. So think of an Uno skip card, and we will just go straight to K, because with I gone, it cannot inhibit the transcription of the K protein. K will transcribe E, which will lead to a dragon with genes for spikes like the Hungarian horntail. In the third case, both I and E have been removed, so going through, S transcribes P, which inhibits I, but since I is not there, it cannot inhibit K, thus K will be transcribed. However, E is absent, and without E, we cannot get a dragon with developmental genes for spikes. Think of it like a link of chains with the last chain link broken that represents E protein. Without the last link, it cannot tether the space in between one object and another. Thus, we do not get the end product of spikes and instead get a dragon like Toothless with no spikes. In the fourth case, both S and K are removed, thus with the S gone, it cannot transcribe the P protein to inhibit I. Again, think of an Uno skip card, in this case because we skip straight to I. I would then inhibit K, but since K is missing, it cannot transcribe E anyway. Thus, the developmental gene of spikes is not transcribed and that dragon will not have spikes. Now one might ask, how come a dragon can have spikes on the skin and no spikes? Shouldn't all cells have the same genes and thus all transcribe spikes? Actually, the reason that the dragon in this picture has spikes in some places, but is smooth in others is due to cell differentiation, specifically methylation in this case that acts as a gene silencer. Methylation of the histone tails can decrease transcription and prevent some transcription factors from binding to the DNA, thus repressing the expression of a gene. The DNA wraps around the histone so tightly, though it has been exaggerated in my drawing, that it silences it, and that spike gene is not transcribed in the cell, making it smooth in some places on the skin and spiky in others where it has been transcribed. So say, for example, this happened to the E protein, and it was regulated by methylation. The E would not be transcribed, and thus one would not see spikes as a phenotypic characteristic. Thanks for watching. Here are my references.